Morning, everyone. Welcome to day 2 of Acharya Red Connect 2025, a premier online webinar series dedicated to advancements in medical imaging, organized by Department of Medical Imaging Technology, Acharya Institute of Allied Health Sciences, in association with Indian College of Radiological Technology. I'm Devlakshmi, your host for today. Without further ado, let me introduce you all to our speaker for today. We have with us Miss Monika Reddy, Assistant Professor Medical Imaging Technology, Department of Allied Health Sciences, Ramaiah University, Bangalore, Karnataka. Ma'am has 1 year of clinical experience and 6 years of teaching experience. Ma'am has been invited as a resource person and guest speaker by several institutes on various interesting topics and advancements in medical imaging. Ma'am has published a paper, Influence of Lumbar Spinal Canal Dimension on Neurological Claudication Symptomatology, a case control study. We extend a warm welcome to you, ma'am. I request you to kindly begin with this section. Yes, ma'am.
Yes, that is
Yeah. So without any further delay, we'll start our session, which is molecular imaging, especially in neuroimaging, in Alzheimer's as well as Parkinson's, how it is beneficial and what types of molecular imaging are there. Let's dive into it and find out. So usually molecular imaging, the term itself stands for imaging done for molecules or observing the molecules to delineate any kind of pathology. Especially here, we are using it for neuroimaging, where we are concentrating on Parkinson's as well as Alzheimer's. So this is in recent years, it has gained utmost importance in a target. It is not on the symptomatology. We are diving into it. We are going deeper and deeper into diagnosis, into cellular level as well as into subcellular level and even molecular level. That's where this imaging associated with molecules has been developed. And furthermore, we have classified molecular imaging. Traditionally, molecular imaging stands associated with radio tracers, which is done by nuclear medicine people. But in recent advancements by continuous development in radiology and evolution of radiology, we also have been taken that molecular imaging specifications. What do they want to see in molecular imaging? They want to observe molecules. Some types of molecules are responsible to generate some kinds of pathology. So identifying that, what kind of structural changes are happening with the elevation or depletion of certain molecules have been absorbed, and those structural changes with molecular abnormalities, either elevation or depletion, has been noted, and we will see that structural changes depending on molecular level under non-radioactive sources, under radiology, not under nuclear medicine. That is what molecular imaging with radio tracers and without using radio tracers. One is invasive and the other one is non-invasive, non-involving ionizing radiation. Key non-radio tracer molecular imaging techniques involves optical imaging and MR-based techniques and ultrasound-based molecular imaging. But for those, we require some kinds of probes or some accessories which take us to or to obtain a positive results in these imaging techniques. Those are called molecular probes. So in optical imaging, again, we have fluorescence imaging, bioluminescence imaging, near infrared imaging. So all these are other types of techniques under optical imaging itself. So these also observe molecular processes in cells as well as in tissues. But what do they use to achieve to that particular imaging are called molecular probes. In MR-based techniques, we use nanoparticles as contrast agents. We don't use some other drugs, but still we are in research to use much more effective contrast agents where the contrast in MR weighted images will be modified according to the delivery of the material. We have other techniques for chemical exchange saturation transfer technique, hyperpolarized and spectroscopy technique. All these techniques, DTI, all these are under a subcategory of MR-based techniques for molecular imaging, functional MRI as well. So all these with their intrinsic properties adapted to the molecules, follow the water molecules in the certain areas, shows us enhancement in the images. So level molecular changes has happened. Ultrasound based molecular imaging includes contrast enhanced ultrasound as well as photoacoustic imaging. So by using laser and delivering ultrasound and laser via optical imaging, we will reach this ultrasound based molecular imaging. Concentrating on MRI more in this particular presentation, not on other modalities. So what are the advantages of this non-radio tracers? Radio tracers we use under nuclear medicine. We are all clear about it and it is in practice. But here, without the use of radio tracers, how can we evaluate that this is Alzheimer's, this is Parkinson's, depending on some molecules elevation. But the sensitivity varies with nuclear medicine imaging as well as molecular imaging under MRI. One benefit definitely when we compare these two modalities, there is no ionizing radiation exposure and it has a high spatial resolution compared to the nuclear medicine imaging and long-term tracking of molecular events suitable for repeated imaging in the patients as there is no radiation exposure involved. What are the major challenges we may see? As we know, there is a lower sensitivity when it is compared with PET imaging or SPECT imaging, and there is a limited depth penetration, especially in optical imaging, a need for advanced contrast agents in MRI. The contrast agents whichever we are giving with specific. So as I have mentioned already, in molecular imaging, the agents whichever we are using to trace out or to alter the imaging are called molecular probes. Molecular probes detect, track, and visualize specific biological materials or microstructures and processes in the body at cellular level, which in return enhances the signal intensity in MRI. So these are categories of molecular probes which we use according to the other imaging modalities. Fluorescent probes in optical imaging, MRI contrast agents in magnetic resonance imaging, radio tracers in obviously PET and SPECT, chemical luminescent and bioluminescent probes, and photoacoustic imaging probes in photoacoustic imaging. So these are the various radio tracers we use for radio tracer influenced molecular imaging. which is a gold standard technique and which is in practice. So you can see different types of radio tracers being used depending on the modality. In that also, again, PET is there, SPECT is there. For PET, we use fluorine, constant, gallium, and carbon. So these are the tracers which are in use commonly for PET imaging, and whereas iodine will be used for SPECT. And there is a undergoing alpha synuclein, especially for still the tracer is being developed, and it can be told as a future biomarker. So you can observe what is the target of imaging target, and what is the different radio tracers being used, and what is its clinical utility. So how to differentiate Parkinson's disease with tremors? We use iodine under SPECT, and if it is being used under PET, we will use fluorine. And dopaminergic function for to look after disease progression, and alpha synuclein, which is still under research, which will be setting a future biomarker. Just by giving that, we will get all other these clinical utility in delineating the Parkinson's disease, especially neuroinflammation, in, in order to look after for microglial response by using carbon 11, and for Alzheimer's as well. We have beta amyloid plaques, which is a target zone where we use 11C or 18F fluorine for PET imaging in order to look after for early diagnosis of Alzheimer's. And to stage the disease, we use 18F, and targeted region will be tau neurofibrillary tangles. Alzheimer's disease. So what is the cause? There is no cause except the degeneration until or unless there is a traumatic brain injury or vascular disease. So Alzheimer symptoms. So you all must have known already what are the symptoms of Alzheimer's. Can anyone respond in chat? Good. Memory loss. So it is a minute, at early stages it starts with memory loss, and eventually they will struggle with their daily lifetime to remember. So at short duration of their lifetime will be lost from their memory, and then later further they will not be able to cope up with daily activities that they were doing. For this, what is responsible? The plaques, beta amyloid plaques, which is present, or the deposition developed in the brain tissue. And then tau tangles. Tau is a protein. The deposition of it within the brain produce some kind of tangles where it makes the tissue unhealthy, the neurons unhealthy and dehydrated and not functioning, eventually necrotic. Further, it goes to neurodegeneration, complete loss of control in the nervous system, and then cognitive decline. Recognition will go down, and then they'll not be able to remember few of their life events, and so on, so on. So this is how usually a healthy brain looks like, fresh and a healthy neuron. Whereas when you compare it with Alzheimer's, there will be a notable shrinkage in the tissue of the brain. And you can see there is spaces in between the brain cortex as well. Whereas in the brain cortex, there is no spaces in between, only there will be gyri and sulci. But here, there will be huge spaces in between this sulci. And also the plaques which we are speaking about, beta amyloid plaques, the deposition in between the neurons, and then the tangles in the neuron. So these are the tangles where the healthy neuron is being shrinked. So this is what it is happening behind the scenes in Alzheimer's, the structural changes in Alzheimer's, just because of the deposition of the protein. So this is how the imaging varies, whereas for healthy brain, the ventricles, everything will be proper, whereas here in Alzheimer's brain, the ventricles will be enlarged, which affects everything in the brain. The minute changes in the brain also affects your thinking process, language, or it shows some kind of symptoms, developmental symptoms in your entire lifestyle. So cortex has been shrunk. The size of the healthy brain, compare it with the Alzheimer's patient. The entire tissue itself has been shrunk. So how the disease progress, it starts with without showing any symptoms, early stages, and the duration of that without symptoms, with small event loss from the memory. We will think at times, oh, this might have happened to me, but we will not be sure. Such kind of temporary memory loss and a short period of our life event was gone, and we will not be bothered about it because it doesn't make much difference. It is not so important to us. But then, as it grows, for if we fail to deal with it or treat for it immediately, then immediately after 7 years, it will take to develop and to damage the further tissues. So here the disease will be spread to lateral and temporal zone, lobes of the cerebrum. And it will take another 2 years to progress it to frontal lobe. So entire tissue of the brain has been damaged and it reaches to occipital lobe as well. So as the disease progressed from lobe to lobe, you can see the symptoms variation. So if it stays in medial temporal lobe itself, you will be developing short-term memory loss. And then after the progression, after 7 years, after its progression to lateral and parietal lobes, you will be having difficulty in reading, and then poor object recognition, poor direction sense, left, right, you will get confusion, you will start panicking with the roads and directions. Then after that 2 years, after as soon as it spreads to frontal lobe, you will have a poor judgment. You will start doubting yourself in any kind of judgment, in colors or in any other daily activities wherever judgment is required. You will start impulsivity. You will go into defense mode because you are not able to judge it properly. You will enter into defense mode, and very short attention. You will a fraction of second, or if someone disturbs you, you will forget about the previous work whatever you are doing. So such kind of deviations will happen, and then you will which affects your daily lifestyle. And the final stage, you will start developing the visual problems as well. Anyone, have you ever sorry, anyone watched this movie? No. Yes. What movie is it? Those who have responded yes. So in this movie, there is a beautiful depiction of that Kishkinda. Yes, good. Kishkinda Kanda movie. It is a Malayalam movie where they have beautifully depicted that Alzheimer's and its progression, how the person will cope up with it, how hard it is to accept that he has Alzheimer's. They have beautifully portrayed the entire scenario or scenes over the Alzheimer's. So definitely those who said no, please try to go and watch the movie. It is a murder mystery though, but it is all depends on Alzheimer's, how the old person will try to cope up with his Alzheimer's without people getting to know. Okay. So next, Parkinson's. What is Parkinson's? What is this difference between Alzheimer's and Parkinson's? So in Alzheimer's, you have a huge memory loss and which affects your daily lifestyle. Whereas in Parkinson's also, you will develop cognitive symptoms and as well as motor relative symptoms, which delineates your ability to perform any of the daily activities. Here, entirely your brain is confused state in Alzheimer's, but whereas in Parkinson's, it progresses to motor nervous system as well, where you will not be able to stand properly, sit properly. You will have tremors while holding anything. You will lose your grip in holding even a pen and to write. So these kind of daily activities will be suppressed. And the main reason for it, there is a dopaminergic neurons, especially in substantia nigra, where the degeneration will happen. So you all know dopamine is how important it is. What is a hormone dopamine? So it controls all your mood, motor control, everything. So there is a huge depletion, degeneration in that particular neurons in substantia nigra, which is responsible to hold that neurons in this particular area. So this area will be, usually it is hypo, and then it will be hyper intense in case of Parkinson's. So this is full of dopaminergic neurons in a normal neuron, whereas diminished substantia nigra will have a, it doesn't have any, you will not respond or you will not be able to show your anxiety or depression or even your mood, you're happy, you're sad, nothing can be seen in your face just because of this neuron depletion at substantia nigra with dopamine neuron, which affects your entire daily lifestyle, as well as these are the main signs and symptoms that you have Parkinson's: tremors, slowness in the movement, abnormal slowness in the movement, rigidity while holding or losing grip while performing any of the activity. Handwriting will vary. You will, because of the tremors while holding the pen, you will not have a grip. So, depression, anxiety, and then you will, even if you are happy, you will, your face will be sad. You cannot express that you are happy in your face. So that is also apathy. That is called apathy and insomnia, loss of sleep. But for this prevention and treatment, exercise, workout, and few medicines might help, and surgery as well will be done. And surgery will be done with deep brain stimulation. So there will be an implantable device which they will keep, and it keeps generating the dopamine hormone to your thalamus region, which is fixed into the thalamus region. So this keeps delivering that into the deep brain so that you will perform or your some of the activities will be reversed back through the surgery. So major biomarker both Alzheimer's and Parkinson's is tau protein, which is an important protein, and the depletion of it, the deposition of more and more of this protein into neurons will be leading to neurodegenerative diseases like Alzheimer's and Parkinson's and so on. So the primary MRI sequences for Parkinson's disease: T1 weighted imaging, T2 weighted imaging, susceptibility weighted imaging, as well as neuromelanin sensitive MRI. This is one of the technique, and DTI, as well as functional MRI. So you can see this in the anatomy where the dopamine group is traveling in substantia nigra, and substantia nigra has three parts where it is divided into pars lateralis, pars compacta on upper zone, and pars reticulata. So below that, and in sagittal, you can appreciate the substantia nigra, and here in coronal as well, just beside the hypothalamus. So what are the key findings which are responsible? How we can use this molecular imaging without seeing the molecule? How we can differentiate that this is Parkinson's disease, this is Alzheimer's disease. So in Parkinson's, that early biomarker is substantia nigra, where it will be the ratio of substantia nigra will be checked. The structural changes in the substantia nigra will be seen to evaluate whether it is a Parkinson's in staging it early. So there are further advanced techniques. High resolution SWI can be performed, and spectroscopy to see dopamine metabolism and its level of dopamine in the spectroscopy. What is the ratio of it in the graph? Is it elevated or depleted? By evaluating that, we can confirm whether it is Parkinson's or any other traumatic brain injury. So neuromelanin. So this is a advanced technique in MRI itself, NM, neuromelanin sensitive MRI, which is used to visualize the neuromelanin in substantia nigra as well as locus coeruleus. So which is neuromelanin is a dark pigment which is found in catecholinergic neurons, particularly dopaminergic neurons in SN, pars compacta. We have seen the parts of substantia nigra, right? So in that pars compacta. So this imaging is crucial for detecting neurodegenerative diseases, especially like Parkinson's. So how the contrast varies in neuromelanin deposition. So this is substantia nigra, where the signal intensity will vary from healthy substantia nigra to neurodegeneration substantia nigra. So there will be increased signal intensity in case of healthy, and whereas reduced signal intensity in neurodegeneration studies. So these are neuromelanin MRI sequences, T1 weighted TSC sequences, magnetization transfer sequences, as well as high resolution under 3 Tesla and as well as 7 Tesla. So these are primary MR sequences for Alzheimer's disease. Whereas there we have seen susceptibility weighted image, T1 weighted, T2 weighted. Here also we have T1 weighted, T2 weighted, and DTI to evaluate microstructural changes depending on the molecular level, which happens with the molecular changes, and arterial spin labeling as well as resting state functional MRI. I guess DTI study is included in furthermore webinars. So kindly concentrate. That will be useful in knowing how to perform DTI and what is a DTI software, how does it work. The major key findings: hippocampal, to evaluate hippocampal and medial temporal lobe atrophy, we use T1 weighted sequence, white matter hyperintensities in T2 flare, as well as hypoperfusion in parietal and temporal lobes in arterial spin labeling MRI, disrupted functional connectivity in dopaminergic neuron with functional MRI. Resting state functional, RSF MRI. It is resting state functional MRI. You are not delivering any of the in functional MRI, as you know, we can give the sensory instructions. So that and all is not necessary. In resting state, we can evaluate this through functional MRI. So advanced techniques included amyloid and tau imaging. As we know, beta amyloid neuron is a protein is the response. The deposition of it makes a neuron unhealthy, right? So these, by using a hybrid imaging PET MRI, by using these two proteins, we are able to check for how much deposition has happened in that neurons, in that tissue. And MRS, so that we can see N-acetyl aspartate versus creatinine. The ratio of N-acetyl aspartate and creatinine will be checked. In case of MRS, there will be a decreased value of this ratio. So here you can see NAA as well as creatinine is elevated. I didn't find a graph which shows the depletion or the reverse graph of this particular pathology. So just to show a spectroscopic graph, I have kept this. So you can see various n number of metabolites are able to be seen in spectroscopy. So what is the comparison between radio tracer versus non-radio tracer? So we have seen the advantages. In case of invasiveness, it requires radioactive tracers, where it is non-invasive and no need to inject until or unless there is a requirement of contrast media. Otherwise, we doesn't require any much contrast also with the further DTI and MRS. Molecular targeting, this is very specifically being done to a specific protein, whereas we are checking the microstructural changes depending on the metabolites. We are not seeing the metabolites directly, how the functioning, how the transfer is being done, nothing. We are not able to check the complete physiology, but here indirect imaging through microstructural changes by metabolites. Resolution, high molecular specificity, and for depending on molecular level, it is low molecular specificity. And for clinical use standard, it is already a standardized gold standard protocol under radio tracer based using PET or SPECT for Alzheimer's as well as Parkinson's. But these are emerging techniques which doesn't involve radiation. So these are my references. Hope it is informative to you all. If any questions are there, we can take now.
If anyone has any questions or doubts, ma'am can answer it for you. You can post your questions in the chat box.
So someone wants to see the references. Yes, I will. Molecular imaging improve the treatment of PD and AD. So there is no treatment with imaging. We can give, we can find it at the very early stage by performing this molecular imaging, and then we can start with the treatment by providing drugs. And without providing drugs, there are cognitive health therapies, mental health therapies that we take in remembering it further and further, or by maintaining a notes about yourself. So all these are therapy techniques which will be provided by psychologists. And motor sensory, they will, as we mentioned in Parkinson's, For tremors and all, they will just do that surgical implantation device. Controlled hormones will be generated whenever it is necessary, so that it will help you for some extent in maintaining your daily activities. How integrating molecular imaging in MRI is better than conventional MRI? Definitely, there is no much visibility of metabolites in conventional MR techniques. We have a restriction in evaluating it only if it is correlated with the biomarkers. Then only we can consider that it is Alzheimer's. We can confirm the disease. Otherwise, we cannot. Do this generation completely affect our brain? Yes, of course. Yeah, I will share it to the host. You can collect it from the host. How to determine it by ultrasonography? There is something called as photoacoustic imaging using the sonography, which is again my newsletter published in Alara magazine. If you want, you can go through it. Thank you.
Any more questions?
I guess there are no more questions. Thank you.
If there are no further questions, we can wind up the session. Thank you, ma'am, for sharing your knowledge with us on how the smallest molecular changes can be detected in case of Alzheimer's and Parkinson's disease, how the diagnosis is made, and how the damage within the brain can be checked for. It was a very fascinating topic, and I'm sure the participants grasped a lot of information from your session. Thank
Sure.
you. Thank
Thank
you
you.
once again, ma'am. Thank you so
As
much.
many of the students are requesting for slides, I will share it with you or Swapnil, someone,
Okay,
so
ma'am.
that you could share it with the students.
Okay, okay.
Or you
Okay,
can
ma'am.
even share the recording sessions.
Yeah.
Okay. Thank you.
And I thank you all the participants for your active engagement today. Before we conclude, let me briefly inform the participants about the next session tomorrow. We'll have with us Mr. Prakash Umbarkar, Executive Senior Radiation Therapist, PD Hinduja National Hospital, Mumbai, Maharashtra, who will be talking on recent advancements and emerging trends in radiation therapy and imaging. See you all at tomorrow at 11. Until then, stay safe, take care, and a great day. Thank you.